Hello, it's Julia from STEM Zone. It's Monday night and it's biology night. We're having a bit of a chat here about biology, in particular in the BCE course. So I'm just double checking that I've set things up correctly. That looks pretty good. And I think we are live. So today I'm talking about the Year 11 course, the Unit 1 biology. And just while I check my phone, so at the moment, you guys have finished talking about crossing membranes, movement across membranes into and out of cells and things like that. You've finished talking about your organelles. And moving along the course now, you should be talking more about reproduction as a whole and cellular processes leading to cell division. So tonight we're just going to have a brief chat about different types of cell division there that you might be talking about in class. And you just let me know via the chat if you guys have any questions while I'm doing this, and it'll be uploaded. You can watch it later, and hopefully I can offer you some assistance with some of those concepts. So let me just get onto here, share my screen. It does always help to have pictures and PowerPoints and things, so that's why we sort of have those to support our Facebook Lives. So today specifically, we're talking about prokaryotes and eukaryotes. Now, when we talk about reproduction, often we might think about sexual reproduction between two different organisms with two different genetic contributors, two different parents, contributing to the next generation of organisms. But at the moment in biology, Unit 1, you guys will be sort of looking at the asexual reproduction side of things. So that is reproduction without two contributors, two genetic contributors. So basically in asexual reproduction, you've got one parental organism producing offspring in the next generation. They're genetically identical to each other, those offspring. So every offspring that parent will produce asexually is identical to each other and also identical to the parent themselves. So asexual reproduction is seen heaps and heaps of different examples out there in nature. I'm a bit of a plant lady, so in particular, I know, in terms of plants, we can take propagations, we can take cuttings from plants, put them in some water, let them develop new roots, and then go and plant that propagation into soil or have it as a hydro plant. And that's basically, you've reproduced that plant and created a new organism. It's the same sort of thing happening with rhizomes and things. Some of the plants you might see, they have roots that grow underground and they spread their wings, and little new offspring, new plants would pop up along the root system and come up above ground. So there's a lot of examples in the plant world of asexual reproduction, obviously. And also in the animal kingdom, you've got like different lizards that particularly, there's a lizard where you've got only females of that species and they reproduce themselves. You've got the hydra, which is a really famous example that is used in class a lot, which is an aquatic organism that basically grows a little bud out of the side of its body. And as that develops, it'll develop till a point of maturity where it can survive on its own, and then it'll break free from the parent. So you can see that green diagram there of the hydra. So there's lots of forms of asexual reproduction, but specifically you guys are talking about prokaryotic cell division, so cell division in bacteria, and that's known as binary fission. So I'll talk to you a little bit about that, and then we'll compare that with our eukaryotes. Before we continue with the binary fission, there are some major advantages to asexual reproduction, and there's some disadvantages too. So just in terms of being able to see that on my screen. So if you're thinking about advantages of having one single parent creating the next generation of offspring alone, that process is really fast. It can be really fast. So you can grow a population rapidly. If you think about bacteria, prokaryotes, when you say you ingest, you've got some food that you didn't reheat quite properly, and you have that meal and you've picked up some single bacteria cells, right? Or say you've picked up a little colony of like a few hundred bacteria cells. Over time, over not much time at all, those individual bacteria that you've ingested will multiply, will divide cells into more and more cells. And the process of division in prokaryotes can take, say, 20 minutes. So every 20 minutes, you've got double the amount of cells that you originally ingested. And that's how we can see sort of food poisoning come on within a few hours of having just ingested something. So population growth can be really super rapid. Another advantage is the fact that you don't have to waste time and energy finding a mate to produce offspring for the next generation. You're self-reliant, you're an independent person, individual organism, and you don't have to spend time away. And if you live in isolated areas, there's not many mates around, and you're going to be competing for those. So that's an advantage if you don't need to actually reproduce with a mate. Again, it's about the speed of reproduction too. So if there was a sudden reduction in a population and we had like a wipeout, and half the population was gone, if we're reproducing asexually, often the population replenishes a lot faster. You're getting more organisms a lot quicker. So there's a lot of advantages to reproducing asexually without a mate. However, there's one pretty big disadvantage to this. There are a few others, but the one major disadvantage to asexual reproduction is that there's no genetic variation in the population. So if I'm a single organism and I'm basically cloning myself, I'm making copies of myself, genetically we're identical. So that might be great if I'm flourishing in my environment and I have all of these great adaptations that allow me to survive in this particular scenario, this situation, this climate, this altitude, whatever it is. And I'm making little mini-me's, little clones of myself that are all also really well equipped to survive in that environment or those conditions, and that's great. But if there is some kind of change, if there's a bit of a disease, a plague or something that one of us is susceptible to, then because we're all genetically identical, often that means that all of us are susceptible to that disease or to that particular environmental condition, something like that. So without variation amongst the population, if we're all identical clones, then that's great if the environmental conditions never change, which is pretty much impossible. But if there is sort of a change or an unfavourable sort of situation in the environment, then you're going to see most of those organisms impacted in the same way. If you've got variation, you're going to have some that can withstand that disease, they might be resistant, or some that can withstand that huge temperature fluctuation or change, because of their fur and things like that. If you've got variation, some of you are going to survive and you'll be able to reproduce and grow the population again. But if you've got no variation, that means that a lot of you are susceptible. Most of you would be. So think about that as you think about reproduction. But generally speaking, you guys are focusing on, in terms of prokaryotes, focusing on binary fission, submission. So here's just a simple image of a bacterium, one bacteria cell. Some of the features include obviously a cell membrane, but in bacteria, they've also got a cell wall. It's different to a plant cell wall. It's made of different material, but they do have that extra rigid protection, that layer around them. It's made of peptidoglycan. It's not made of cellulose like a plant cell wall. So they are different, but they've got that double layer around them, which is protective. A bacterium also has circular chromosome as their main DNA, and they've got other smaller sections of DNA called plasmids. That is covered a lot more when you guys continue on into year 12 as well. They look at what those plasmids are for and how we can use them and things like that. But generally speaking, the prokaryotes, they don't have membrane-bound organelles. They have that circular chromosome, they have some plasmids, which are a few extra genes. They have ribosomes. They have that cell membrane, that cell wall. They don't have other membrane-bound organelles. So we sort of think of them as like a simplistic, a more simple organism. But in fact, they're just as evolved as us. They've been around a lot longer than us. They've survived environmental change incredibly well. So I wouldn't exactly call them simple creatures. They have less structures than we do, less compartmentalization than eukaryotic cells. They're amazing. So, general structure of a prokaryote, if this prokaryote is going to go and divide and split to two new cells, one of the main things that has to happen is that the two daughter cells have to end up with the same features, like DNA, same genes. They both have to have a copy of that bacterial chromosome, and they both have to have some plasmids, some ribosomes, etc. So the general sort of process we call binary fission. So fission is separating, fusion is coming together. So binary fission is when you've got your sorry, I'm just got a notification. You've got your parent cell, your original cell starting there, your nucleoid, which is that circular chromosome, that piece of DNA that's in a continuous, like a circular shape. Obviously, it's all coiled up in that image, because it's a brilliant long piece of DNA. But that's, in contrast to eukaryotes who have linear DNA. So our chromosomes are made up of long singular pieces of DNA that do not then join together to form a cell. Anyway, so we've got our nucleoid there. You can see the cell wall. You can see the membrane there. Basically, our DNA, not our DNA, their DNA is replicated. So we copy that circular chromosome, make an exact copy of that, so that there's two there, one for each daughter cell. And the cell membrane will elongate, which means they just build more and more phospholipids and, extend that membrane. There's then like a bit of a plate that forms because they have cell wall. It's a bit of a plate that forms in the middle there as the two new daughter cells begin to separate. And eventually what they'll do is they will just separate away, and that cell wall, the cell membrane, will close the gap basically and seal it into two new daughter cells. They're not called daughter cells because there's anything to do with two X chromosomes or anything gender related. We just use that term in biology to say we've got a parent cell and daughter cells are produced. We don't have son cells or boy cells or anything like that. So our daughter cells are produced, demonstrating that there's the offspring in the new generation from that original parent cell. Cool. So it's binary fission. It's pretty simplistic. You copy the DNA. We make sure that there's enough material to go into both cells. So there would be ribosomes being replicated, and you're making more with that cell membrane and the cell wall. You're elongating those walls, and eventually it will pinch off and form two new cells. So in prokaryotes, it's a lot less involved because they have less compartments. They don't have membrane-bound organelles and all that sort of stuff. Awesome. Okay. In eukaryotes, it's a bit different. You and I are eukaryotes. Our pets at home, our plants at home are eukaryotes. So we have a bit more of a complex system with eukaryotes. And hopefully in class you have talked about the life of a eukaryotic cell before you jumped into its reproduction, reproductive systems. So we talk about the life of a cell being called the cell cycle. So it's like a life cycle of a cell. Now, a cell is born, so it's created from an existing cell that existed before. It's born, it's created, it then grows and develops and does its thing. If it's related to the digestive system, its job might be to secrete some enzymes for digestion, like it's doing its job as it's growing. And some cells then will need to divide and keep reproducing, and so they'll go through the cell cycle further, and some of them won't. So I'm just going to break that down a little bit now. So, for example, you can see on the diagram here we've got two cells here, two daughter cells on the right-hand side. So let's say that this is the start of our cell's life. The cell, you've gone from having one cell that's split into two smaller ones. So part of the next phase of its life is to grow. It's called growth 1. It's to enlarge, it's to produce more organelles. and it's also to function as a cell, to perform its function. So like I said, if it's a digestive system cell, it's got jobs to do. If it's a heart muscle cell, it's got jobs to do, and that sort of thing. So during growth 1, the cell is growing and it's building its cellular contents and it's performing its daily functions. Now, some cells do not replicate. Some cells, for example, heart cells, you're born with certain heart cells, and once that organ stops and is done sort of developing, those cells don't replicate any further. So if you damage some of your heart cells, like this is why heart disease is such a problem, heart attacks and things like that, if you damage some of your heart cells, you don't replace them with new heart cells. That damage sort of stays there. And we work around that. So there's lots of different, treatments, drugs, surgery, different treatments that we've been working on. But that's because you don't make new heart cells. So in that case, the heart cells would go through G1, they'd be in their growth 1 phase, and basically they would then enter into G0. It's the arrest, cell cycle arrest, or stopping of the cell cycle. It's not going to progress any further. It's going to enter into G0, which is like a dormancy almost. It doesn't go any further, and it will live out its life in that phase. So it'll still continue to act to be a heart muscle cell, for example. It's going to do all the jobs required, but it has a finite lifespan, and it's then when it dies, it's obviously that's it. It's not going to divide into 2 new cells at any point. Another example might be your nervous system, your neurons, your nerve cells. They can repair themselves, but after you reach a certain point, they no longer replicate. You're not making new nerve cells anymore. So again, when there's damage to nerve cells, sometimes there's repairs that can be made, but if things are severed or that sort of thing cut out, then there's no replacing those with new nerve cells. Same with your brain cells. So some cells stay in G0 and do not replicate. If, for example, you're a skin cell, and say, if you're a little bit uncoordinated like I was growing up, and you might be fully active as well and very sporty, so I was falling over a lot, scraping skin off my knee, etc., right? There's not a permanent gaping hole in my knee. Those skin cells that I cut off or shed off or whatever have been replaced with new skin cells. If you cut yourself now, there's a gap in your skin, I guess. That gap doesn't stay like that. Cells around that cut clean up the mess. They get rid of bacteria and stuff like that, and they fill the gap with new skin. So that's what we're talking about here, is that if a cell is going to go and continue on its journey and end up dividing and becoming 2 new cells for growth or for repair, then there are different stages to go through in the cell cycle. So once we pass our G1, if we're a cell that's going to divide into 2 new cells, we've then got to make sure we've got enough of everything to do that, to create 2 identical daughter cells that have the same amount of DNA, same genes, all right, and enough contents to survive and that sort of thing. So S phase is synthesis phase. So once we're ready to go and divide, we're like, okay, we need to replicate our DNA. So S phase involves replicating every single chromosome in our eukaryotic cell. So there's 2 copies of every chromosome. There's checkpoints along the way, which we'll talk about later in another video, another chat. So basically, S phase, at the end of S phase, we'll double check that our DNA has been copied correctly. If so, we will enter growth 2 phase. So there's more checks along the way before this cell goes from 1 to 2. Any errors in that replication, any errors in copying the DNA will be detected. It's crazy, I know. We've got enzymes, we've got proteins in our cells that basically go over that DNA and can detect any errors if there's missing nucleotides and that sort of stuff. we'll make any repairs before mitosis, before we're going to divide our nucleus and divide our entire cell body, right? So that's what we mean by cell cycle. There's there's a number of phases that the cell will go through, and will check along the way before it actually divides and becomes 2 new cells. So in your course, you'll be looking at the cell cycle and different checkpoints along the way, and what can go wrong along the way in the cell cycle. Things that can lead to cancer and stuff like that. Like, you've got errors that can happen in this process that could lead to cancer, but that's also why we've got checkpoints. Again, that's for another time. So I'll just keep going on. So basically today was about cell division. So we talked about binary fission in prokaryotes, where there's that replication of DNA, then that separation into 2 new cells. Now we've got our replication in eukaryotes. So in you and me, and in our plants and things. Hopefully you've had access maybe to some microscopes in your time. This is an image of growing onion roots. So the tip of an onion root, and it shows cells at different stages of division. And all of those beautiful chromosomes are highlighted. We've got, cells that we've got a very dark nucleus. We can't see any detail within that. Then we've got cells here where there's clearly long strands there being pulled away from each other. The cells are not all in sync as such. So they're not all about to go, all right, let's divide, bang, and then everybody's doubled, right? They go through their cycle. They've got different triggers and things. So you're going to see cells at different phases. Here at the top sort of left area, you've got new cells that have just formed, right? So hopefully you've had a chance to see this. This demonstrates the idea of the onion root, the root of the plant growing and elongating because new cells are being made. So when an organism grows larger, it's not because the cells are getting bigger. It's getting larger because we're getting more cells. So that's what we're talking about here. Bit of a GIF there just to show the idea of one cell undergoing some processes, obviously super sped up. But mitosis in eukaryotes, like, you've got this one cell that undergoes the processing and then it becomes 2. So all cells that exist come from pre-existing cells. I think that's mind-blowing too. All right, so mitosis, I'm just going to step through the phases. You would have drawn this in class, would have talked about it a fair bit. This is just sort of a reminder of the key areas. So the bottom left here, after S phase, what we've got is duplicated DNA. Now, chromatin refers to the DNA that's unraveled. It's all loose and really accessible. It's not in chromosome form. Now, if you think about replicating DNA, we have to make a copy of every single piece of DNA. So we have to be able to read the code that's within the DNA. We have to see how the DNA is rearranged to then be able to make an exact copy of that. So if the DNA was all tightly wound up in these chromosome form, you can't access that code. You can't access the order of the nucleotides of the DNA. So during S phase, we see DNA unravel from chromosome form and really be in long chromatin form. Now, mitosis, the ultimate goal is to create 2 new daughter cells that are identical to that original parent cell. And if you think about the idea of mitosis, it is nuclear division. It's division of the nucleus. It's not really cell division. It's specifically division of the nucleus. And what's in the nucleus is DNA. So mitosis is about evenly distributing and dividing up the DNA so that 2 new daughter cells can be made that have the same DNA as that original parent cell. And if you think about, I always use the analogy of pasta. I love pasta. And it just makes sense to me to think about DNA in this way when we're talking about cell division. So think about a bowl of spaghetti, long, skinny strands of spaghetti. You've got a big bowl of spaghetti. Try and imagine separating that spaghetti evenly to two sides of your bowl or two sides of your plate, evenly, same number, and making sure that you've got, like, if there was two long bits of spaghetti, one went on each side of the bowl. If you've got your two shorter ones, then you get two on that side, two on that side of different lengths. Like, imagine trying to separate out a bowl of spaghetti. It would all get entangled with itself. It would be very difficult. You would end up with a lot of unbalanced, errors in your product there with spaghetti. Now, if you were to picture that again with penne pasta, those short, stumpy, nice little movable chunks of pasta, you can imagine that'd be a lot quicker, easier, more efficient to separate out into two groups, into two sides of your bowl, right? Spaghetti versus penne. Maybe you can relate to my analogy. Same goes with cell division and mitosis. Mitosis is the division of the nucleus. So if you had to separate all of that long, stringy DNA to either side of the cell, there would be entangled sections. You'd get breakages of the DNA. You wouldn't end up with it being evenly distributed. So in the first part of mitosis, the chromosomes form. Your DNA condenses into chromosomes, like into your penne pasta, to make it easier for that whole process to occur. So in prophase, that's the first part, is we've got our DNA condensing into chromosomes. And hopefully, the way you're thinking about the phases of mitosis, like these are on a continuum. It's not like this happens, then a button switches and the next phase happens, and then the next phase happens. It's kind of like they happen on a continuum. But as biologists, we need to be able to make sense of the process. We like to categorize things and sort of make it easier to understand the process. So we say that they're distinct sort of processes, but it's on a continuum. So during prophase, you'll see chromosomes condense, centrosomes will be migrating to either side of the cell there on the left here. You've got your spindle fibers, your protein spindle fibers coming out and forming those long attachments. They will then be working their way towards the DNA, and your nucleus is breaking down. Mitosis is the division of the nucleus, right? It's division of DNA evenly into two new cells. If you can't access the DNA and it can't be separated, this is not going to happen. So you have to logically remove the nucleus. That breaks down so that DNA is freely able to move through the cell. So prophase. Then we see the chromosomes. So along different sections, like different areas along a chromosome, you'll have a point where the two sister chromatids are connected. That's called the centromere. The spindle fibers will come along and attach to a chromosome at the centromere. Specifically, it's like, picture a belt around someone, and like when you want to go and pull them somewhere, you're not going to just grab onto their hair or their skin or whatever. You're going to have a secure sort of anchoring point where you're going to get the whole person. So, picture the spindle fibers sort of reaching out and connecting to the belt of the chromosome. And the chromosomes then start to move around because they're attached to the spindle. When you reach metaphase, you have this lining up of chromosomes along the equator, along the center point of the cell. So meta, for me, is in the middle. We're lining up in the middle. We're lining up single file. You can see chromosomes lining up along that middle plate in single file. Both sides attached to a spindle fiber each. That takes a little while, bit of a dance there by the chromosomes, and then you see the spindle fibers start to shorten. And when these shorten, they're obviously going to be pulling whatever they're attached to. They're going to be pulling that to the pole, to the other side of the cell. So this is how we separate our sister chromatids back into chromosome form. Now, hopefully in class you've talked a little bit about DNA in general, structure of DNA, and the difference between sister chromatids and chromosomes, etc. If not, have a look at that, look online, talk to your teacher about that. So we've got anaphase, where we see our sister chromatids separating away from each other and migrating towards either pole, either side of the cell. After that's happened, we should have an even amount of DNA on both sides of the cell. Then you see telophase, where new nuclei start to form. The nuclear envelopes, made of those phospholipids, we get our new nuclei forming around that DNA on both sides. During that phase as well, telophase, you're also getting this cleavage furrow forming. So it's the start of cytokinesis. But like we said, this is like on a continuum. It's not a distinct phase and then you flick a switch and it's the next phase. The cell is undergoing this process. And so over time, you'll see that cleavage furrow. Now, telophase is actually the end of mitosis, because mitosis is the division of the nucleus. But complete cell division requires that last phase of cytokinesis. Splitting, cyto meaning cell, so kinesis, splitting of the cell, separation there into two new daughter cells with the same identical genetic makeup as that original parent cell. So that's mitosis. It's happening all the time in plants, in eukaryotes, the division of the nucleus, and then leading to cytokinesis. So you've got these two complete new cells. It's happening in your skin, in your digestive tract, anywhere where you've got cells that are undergoing like growth and repair. This is the process that gives you those new cells. So just lastly, sorry, if we think about eukaryotes, there's also within that category, you've got fungi and you've got plants. In plants, there, as an example, you probably would have talked about in class, there's a slightly different sort of process that needs to happen because they don't just have that cell membrane where cytokinesis, is my mouse gone? There it is. Cytokinesis occurs and you've got these two new beautiful daughter cells. When you're talking about plant cells, you've also got a cell wall that needs to form. So you can see a slightly different process where you've got organelles within either side of the new daughter cells secreting different proteins and things that are going to form that cell plate. And eventually that will become the cell wall. Once those two sides are sort of formed, that will then separate into two new separate plant cells, new daughter cells. So slightly different, steps there because of the different structure of the cells. But I do think that's it for now. It's great to see you guys come along and have a bit of a listen with me. And feel free to get in touch with us if you've got any questions. Chuck us an email. Talk to us on Facebook, Instagram. We're just wanting to share our ideas, share some of our knowledge on these topics that you guys are covering. So next week for biology, we'll be doing Year 12 again. Classes sort of flow at different rates. So we're doing our best just to make sure we keep up with you guys and we can give you a bit of a revision over here on Facebook. So I'll stop sharing there. Thanks a lot for coming along and we'll talk to you again soon. Carissa will be in tomorrow and Wednesday. We've got some chemistry and psychology coming up. So see you then. All the best, guys.